response. These are nothing to do with the housekeeping response. And so it increases or enhances functionality of those genes which can be affected by, say, heat, acid, all kinds of external factors. Okay? And so the sigma factor always increases the specificity. Okay? And also at the termination, the separation from the DNA, that also helped by this sigma factor. Okay? And so those are already described in the slides I gave you. So read up that information. And this is going forward. This is going to be an extremely important understanding. If you, like I said, in order to understand why microbes showing resistant to antibiotics, right, or resistant to certain phenomena, other factors, and this will help you to understand that better. Okay, if you can regulate this sigma factor function, maybe you will be able to control their physiology, their action, whether it's for disease or for any purposes. And so that's why this sigma factors, knowledge of sigma factor is extremely important. Everybody clear? Okay. Any other questions? Go ahead. Okay. So what is going to regulate, right? Well, that's a good point, though. When we said that there is no nutrient, right? Nutrient starvation, or pH, or salt, any of those external factors, or antimicrobials produced by other microbes, they can also regulate the QS synthesis as well. Okay, so that means that can affect the communication between cells or among the cells. So the production of the QS molecule may be affected depending on what conditions they are in. So if they are in a healthy environment, but there is lack of stress, they may not need to produce it, right? We all agree, when there is a stressful conditions, that situation is very adverse, they cannot survive well. So under that conditions, they would decide that I need to produce, a QS molecule so that I can communicate, let everybody know this is the situation. And so how do we handle or overcome these kind of adverse conditions so that everybody benefits in that community? Okay. Well, so yes, I mean, I guess if you don't like for them to form biofilm, right? And so they are forming biofilm, they are communicating each other also, these molecules. So if you disrupt the QS production, or there is an anti-QS molecule, which unfortunately I didn't tell you all of those, I'm waiting for the last day or day before. We have how to control biofilm formation. We got a lot of information, I'm going to talk about that. Yes, you will be able to control or regulate it. So the organisms which produce it, and if you limit their growth, or if you prevent them from being part of the community, they will be producing less EPS. A number of ways you can control that. Okay. Go ahead.
So does sigma factor only identify that cell is under stress condition or normal condition or any other?
That's one of the major indicators. One of the major indicators. So if you look at you can analyze the sigma factor in a gel, or you can see the proteins which are regulated by sigma factor. If they are increased, then you know their cells are under stress. Make sense? So they will produce number of different types of proteins or protein factors. I can say that acid resistant protein, for example, that protein in response to acid resistance, they will synthesize that one. So if you look at if the cells are under stress, they will make large quantities. If they are not under stress, then you will see their level is pretty low. No, I think that's an indication. This is like if you have fever, for example, if you have a fever and you cannot say which organism is causing it, right? Fever is a symptom of many type of infection. You see this? So that way, so if sigma factor is upregulated, you cannot say that this gene is going to be upregulated. Or if you find a gene product is upregulated, you cannot just say that this is the only thing happening, other things can be happening. multiple factors can contribute to that function. Okay. Anything else? Okay, let's talk about, so let's finish that culture cell model. So we talked about the animal models and organ culture and the cell culture we are going to talk about. Any questions do you have? So these tools I've been mentioning, definitely these are powerful tools for studying pathogenesis, right? I think I'm going to go back and okay. Do you see this? I didn't talk about this. Have you seen this before? Called house shrew. Have you seen that? They're called house shrew. I think many of you may have seen it, right? So at home they would be around, right? They run very fast. So these are not marked. These are pointy. They have a pointy face, correct? And they run very quickly. I've seen it when I was a kid. The last time I think I've seen it, the room I've been in. So I think this one just ran by very quickly, and they run very fast. I've never seen a mouse. They run. Okay, which one? Oh, okay. Go ahead. Okay. Anyway, so that house shrew, usually that's generally don't use for, or I would say not widely used as a model, okay? But I did put it there for there's some reason. One reason is this, is that you heard about the Staph aureus food poisoning, Staphylococcus aureus enterotoxin, they cause vomiting. Do you know that symptom, right? So anytime you have this toxin in food and within 30 minutes to 1 hour, you start vomiting, right? Have you experienced that type of symptom? Some of you, right? Don't deny it. We all experience, especially in Indian food situations. A lot of times we encounter that. Back in US also we had that. So the whole idea being is that this vomiting is the main symptom of that toxin, and large number of people suffer from it, right? Because it's easy to be transmitted to food, because by handling, Staph aureus everywhere, right? And under right conditions, they will produce that toxin. So one of the symptoms of the toxin is vomiting, right? Or emesis, emetic toxin, we call it. None of those animals, except pig, you know pig vomits? Have you seen that? Or if you don't know that, pig is one of those animals like humans, we vomit, right? But not all animals can vomit. And so within the rodent family, none of those rodent models they can vomit, or they can replicate the emetic toxin or emetic effect what we experience. So what they use, actually they use this house shrew. House shrew, this animal can vomit. Okay, so that's why for Staph aureus enterotoxin study, they use this animal model to determine if the toxin you are using is a vomiting toxin or emetic toxin, right? And we have isolated scientists isolated over 26 different serotypes of Staph aureus enterotoxin, okay? And one of the criteria, there may be more toxins produced by this organism, but one of the criteria to be a Staph aureus toxin, make sure they test that using this house shrew. Everybody clear? That is the reason I wanted to point out, because all those other animals are used for different purposes, but this one only used for vomiting testing. Okay, so just I thought it's a fun, because house shrew is very unusual, uncommon rodent member, and we use them for as an animal model. Okay, so we talked about different animals you can use, and we talked about if the animals are not available, you can sometimes use organ culture as a substitute for animals, and then that can help you to study. Now we are going to talk about the cell culture. And cell culture model is very common, okay? And because we don't need to use the whole animal, we are relying on the cells from specific organs or tissues, right? And that will be easier. So there are two types of cells, okay? One is called primary cell line, other is called secondary cell line. And by definition, probably you all know what is primary cell line. Primary cell lines are like if you grind up kidney or grind up liver, right? So immediately you are going to say, you can make a cell suspension of liver cells, okay? Then those cells are called primary cells, okay? And the secondary cells, which are actually a cancerous cells, okay? The problem with the primary cells, they have a short life, right? How long they are going to survive in a flash? Come on, you all know the answer. We talked about it yesterday. Close to 10 to 12 days or 2 weeks, right? That's their lifespan, primary cells, always, if it is not nerve cell. And so that's the general rule. So when you harvest these cells and make a suspension, and you can study these cells. For example, if you're trying to study hepatitis virus or other pathogens like a Vibrio, and I want to use hepatic cell line. The reason I want to use hepatic cell line because these pathogens attack liver cells, okay? And so you can use this cell line to study their pathogenesis, whether it's adhesion, invasion, or cytotoxic effect, or apoptosis, or any kind of damage it may induce. You can study by using those primary cell line, got it? But the problem is you have to use it within that 10 to 12 days or 15 days window, okay? After that they are going to die. The next time you need it, you have to use it again. So that's a problematic, right? And problematic also, you need to have a live animal to harvest those cell lines, right? They want to join the class? No. So for that reason, primary cell lines are not going to be useful. So we need something more permanent, right? And that's when we started using the secondary cell line. And when you say secondary cell line, they are derived from cancer cells, okay? What is the purpose? So that they are called immortal cells. Cancer cells are immortal, right? And so those cell lines, we call it cell lines. They are no longer cells, we call it line. Line, whenever you see this word cell line, that relate to cancerous cells or secondary cell line. Is it clear? So the advantage, they are immortal and you can culture them in the laboratory. As long as you provide them food and nutrient, you can maintain them forever, like cancer. See this? And so, and then what happens, originally there are a lot of this cancer cell line already established. So for example, KECO2, I think we use that in our experiment, or people use that more commonly, and that cell line actually colon cancer cell. Colon, it comes from colon cells. So it's called Caucasian colon cancer or something. That's what the KECO comes. CA comes from Caucasian, means some white guy, person died, and then the next one is the CO, stands for colon, okay? And so that cell line is widely used, but that's the picture of that KECO2 cell line, and this will grow, but it's never going to die unless you provide nutrient, okay? So that's been commonly used. So you don't have to have an animal to harvest this. You can purchase it from the one of the major sources, ATCC, American Type Culture Collection. They always maintain it, okay? And so this is generally what you get. So secondary cells, as you mentioned, they are transformed or hybridoma cells, and they have, if it is a transformed, that means they have a viral gene, so called SB40 or Epstein-Barr virus. These are known to cause cancer, okay? And these genes are introduced into the cells to make a transformed cells. And hybridoma cells also secondary cells, and you can create hybrid cells, right? You have two types of cells, you can fuse them, engineer them to fuse it, especially for lymphocytes, they make that. Whether it's a B lymphocytes, which makes monoclonal antibody, or T lymphocytes also for studying a lot of different diseases. So those hybridoma cells are also called secondary cell line. And they are going to maintain a lot of their physiological attributes, including if they are immune cells, they are going to maintain their immunological properties. So that's the advantage. So a lot of study you can do using these cell lines. And these are some of the examples of those different type of commonly used cell lines. HepG2 is from liver. If you need the larynx cells or lung cells, you can use HEP2. Henle 407, this is jejunal cell or intestinal cell line. HeLa cell, this comes from cervical cancer. The female cervix or cervical cancer cell line is the HeLa one. And J774 is called macrophage cell line. Vero cell, which is called monkey kidney, usually is from African green monkey, their kidney cells, okay, and these are called Vero cells. The Vero cells are highly sensitive to virotoxin. Virotoxin is actually toxin produced by E. coli. E. coli 157H7 or some of those, the toxin which damage kidney function, and we tested using the Vero cells. So the cell line help you to assay for many toxin or pathogenic interaction. The last one is called Chinese hamster ovary cell. So clearly it's ovary cell line. So all of those cell lines has epithelial characteristics. They look like epithelial cell, okay, or the macrophage cells. All those different types of cell line can be used. So now think of all those different diseases, different things we talked about. Essentially you can find cell line to study their pathogenesis, right? Yesterday we talked so much about pathogenesis of sessile versus planktonic cells, right? And what did we use? We used this cell line. Then of course we use the live animal, okay? And so this is how we design our experiment. So if you are designing experiment as a graduate student or researcher, a lot of times we study first with the cell line, okay? Once we know the data, if it works, then next level would be animal model, right? Then we use animal model and we verify, prove that this, whatever our hypothesis was, it works, right? Or you came up with a drug, or you came up with a vaccine, you tested in animal model, and then you found the data. The next level would be human, right? We call it clinical trial. So these are the stages of research, what you see when you see the application in humans. So before it being anything being tried on humans, we go through all these steps. In vitro, cell culture model, then rodent model if you have, then we go through the farm animal model like swine model also used, then we go to human model. Everybody clear? And then after that we say definitely it's going to work or not. This is no matter where, what you are doing, always any kind of research application, everybody use these tools. So these are like a common tools, like your computer in your office, right? You cannot do any work right nowadays without computer. You cannot do biomedical research a lot of times without these tools. So these are powerful tools. Okay, so let's talk about how do we culture them, how do you grow them, what are the specific requirements. So this is KECO2, of course. This is a kidney cell or Vero cell I mentioned, and these are lymphocyte cells. Okay, these are B lymphocytes, but these are actually hybridomal lymphocytes. So if you look at B cells, this is what they look like. They are in suspension, and you can see this nucleus, and then the the cytoplasm, and then the membrane. And all these cells look nearly healthy. This may be in trouble, have some problem. This may be, but generally they look really bright. This is definitely dead cell. So by looking at these cells, you know which one is live or which one is dead, right? And that's the powerful tool you can use. I'll show you some more data. So how do you culture them? Remember, these are all human or animal cells. These are like us, right? And if you think of us, what we need for our growth, okay? So we need hormone, for example, right? We need amino acids, we need sugar, we need all of those components what is needed to survive or to maintain our health. Cell lines also need that. And those specialized media are available. I don't know whether you have any cell culture facility on this campus. Anybody has that? That's excellent. So you've already got good idea how it works. But for those who haven't have exposure to this, just to learn that there are specific requirement for this maintaining cell line. So if you have to have a cell culture incubator. These incubators are specially designed to maintain certain gas level, say like carbon dioxide, which is essential. Even though we depend on oxygen, but we still need carbon dioxide for breathing purposes, right? So anywhere from 5 to 7% CO2 you need. You have to be humidified, temperature controlled, and that there is a water bath or water fan to maintain that humidity, and they will continuously circulate that. And these are usually pretty well controlled, maintained equipment. So these are the first requirement. And when you look at their media or the food, you can buy some commercial media, and they already have these essential amino acids, sugar, salt, buffers. And then the most important item is that called fetal cap serum or serum. And serum is the one which is essential. Without serum, you cannot grow these cells. And if you look at the serum has a lot of micronutrients, right? Serum has hormone. Serum has a lot of growth factors. Serum has a lot of cytokines. So a lot of those factors needed for the growth of cells, and you can find it all in this cap serum. And they use cap serum because it's the young, right? And it doesn't have a lot of immunoglobulin, because when you become adult, our serum or our blood carry a lot of immunoglobulin. And those immunoglobulins means antibodies can attack a lot of organisms, right? Because they are against certain molecules. And so if you have too much of those immunoglobulins, they might attack those cells you are trying to grow. So that's one reason. And then also they have lower level of certain cytokines or growth factors. So for that reason, always you have to use cap serum or cap serum. Okay. And then it has also indicator called phenol red. And essentially when they grow in this cell line, the cell line grow in this media, they're going to utilize the sugar, right, carbon, or they're going to produce byproduct, right, which is acid. And if you have too much byproduct made, that would be toxic, right, because they are all in small flasks. And if you leave it too long, the cells are going to die, and you don't want to kill them. So that's why you have this phenol red indicator called acid indicator. So usually the media color is pink like this, and then if it grows up too much yellowish, then you know it's time to replace the media or do something, otherwise you're going to lose all the cells. So this is how whole thing is made. And fetal cap serum, usually I think the story behind in US we get this quite a bit. Usually the calf the day it's born, usually the next day they will sacrifice this animal. They will collect the blood. And I don't know what they do in India, but in Western countries or other countries, they eat veal. You know what is veal? V-E-A-L. Have you ever heard about it? Probably none of you. Okay, I mean, it's not surprising. Usually it's very uncommon word. The veal is the meat of a calf. Okay, that's why it's called veal. It's very soft, tender. They market that. So veal industry is also a big industry in US. So what they do actually, they don't need also the male calf, right? Male calf are useless because they are using always dairy for milk, right? So those dairy cows giving birth to the male, female, and so the male calf, they usually kill them. And they kill them, they collect the serum, because the serum industry is huge. There is a huge requirement. And at the same time, when they harvest the serum, they will use the meat. And so you can fulfill two markets in that. So the veal market is also pretty expensive market. Okay, at same time they use that serum for this purpose. So I don't know exactly how this industry works in India, because I don't think they do this type of practice here, and commonly people don't eat. Say that again. Just ABS? No, but it's still, that's from the adult cow. Adult, when they slaughter it, they collect that, they use that for ABS. You can use that serum, but for these purposes, you need to have a cap serum. But you can use, like I said, adult serum, adult cow serum after slaughter, but those serum needs to be processed properly, and plus it doesn't have all those factors. So probably they can also import from other countries like US, usually very expensive. That's the most expensive item. So we buy a 500 ml bottle, costs sometimes $1,000. Okay, so now you know how much it's going to cost. It's very expensive. The price is going up. So just to tell you the side story, because where this cap serum come from, why you have to kill the cap, is there any use for it? Anyway, so my goal is to always make you know a lot more, right? Sometimes you use cap serum, but you have no clue where they come from, right? Anyway, so these are some of those media. For if you pick up any of those cell line, the Hubeck called, this is endothelial cell line. And there are some specifications, again you have these slides, right? Specifications of media. They formulate in certain way that this cell line, if you're trying to grow it, you should use this media, okay? So you already have this knowledge or this slide, so you could use this as a reference for future. And I use this also same way. So advantage of culture cells is pretty simple, whatever I just mentioned, and you can simple and controlled model to study host pathogen interaction or toxin activity. A lot of this cosmetic industry also they use this before they go to animal studies, right? How do you know it is a non-toxic? That's the first question. So you can use cell culture model first. And they grow rapidly, so it's easy to use them, convenient. You can try with toxin, pathogens, whatever it is, right? You don't have to worry about. It's not that animal. And then primary cells can be, like I said, you can use primary cells, no problem, but you have to harvest each time you need it, right? You cannot maintain them. And then because it's a primary cells, each time is going to be a different result. So it may not be reproducible, okay? But primary cells are like it, there is a reason for it. Primary cells actually true representation of that organ or tissue you are going to use, because it has a mixed type of cells. Secondary cell line only pure cell, that cell line. And the primary cells you will have say epithelial cell, then you have a fibroblast, you may have some immune cells, macrophages, all those other cell type in that lung, liver or kidney you harvest from. So you can have all of these. So that's the advantage, but there is a disadvantage, right? So for that reason, you have to understand what you need, how you can use those for your research tool. Culture cells are derived from tumor cells. So that's the problem, right? Disadvantage or limitations. So their properties has been changed. They are not natural or normal. Okay, so that is one of the issues. So they may not be genetically identical. Continuous growth will produce mutations. That's our other issue. They will be changing. So you cannot subculture all the time without thinking that they are not going to be changing. So they are going to change sometimes. So that might affect your result. So that's why you have to maintain certain passage number. So you grow them for a while, and then you stop that, then you go back to your original, and then you start all over again. And for maintenance, you have to also have a liquid nitrogen storage tank where you can keep those cell line for extended period of time. Okay, immortalized cells are different from the normal cells. Loss of shape, distribution, surface antigens. Culture cells are not polarized. This is another thing. You understand what is polarized means? When you say polarized means you think about North Pole, South Pole, right? So the polar means in the gut, epithelial cells, some cells are facing, certain properties, cellular properties are always upwards, right? And then certain things at the bottom. But when you grow them in the flask, you don't know which is top and which is bottom, right? Which is our outer layer, which is the bottom layer, right? So you don't have that. So that's the problem, you cannot have that. And so you have to make it polarized, and that means you have to use proper cytokine or growth factor, proper hormone. You can use it to make that happen, okay? And so you have to know that. So I will show you some examples. Yesterday I think I showed you this KECO2 cells with the microfiber, like when Listeria trying to get in. Those are polarized cells. We made them polarized, okay? You can do that. So that's the part of the technique. What else? The other thing you can do, because it's a single cell, you don't have all those other cell types, That's the primary cells you will have. Anyway, you can, with the secondary cells, you can add other components. You can add mucus, you can add other antimicrobial molecules, a lot of things you can add to create or simulate like what it is in the gut or in the lungs or in the kidney. You can play with this. And nowadays actually a lot of people are using stem cell, right? So everybody knows what is stem cell is, right? Stem cell is the cell where all the cells are originate, right? So our bone marrow, for example, that's where the source of stem cells, okay? And so if you run out of certain cells, the body actually replaces those cells using the stem cells. They can differentiate. So yesterday, one of you had a question about the ear, right? Organ culture, right? We talked about, so usually a lot of times we start with the stem cell. So we put the stem cell in that culture media, then we have to use proper cytokine or growth factor, then it can differentiate into whatever the cell type you want. Because stem cell is the original cell, right? And it can differentiate into the type of cells you need. And you do that by adding proper cytokines or growth factors. Got it? So if you ask the stem cell to become called epithelial cell or fibroblast cells, you can make them do that. You ask the stem cells to be immune cells, you can make them too. So you have to give them. So that's what we start with many times. The stem cell research is also extremely important areas of research. Anyway, so how do you assay it? So the measurement of virulence. So of course when you are using animal models, certain way you can assay it, right? Assay them. So these are different way you can introduce your pathogens of interest. And then one of the thing, if you are looking for death or life, it's called lethal dose 50. Are you following this part? So like if you want to know the pathogen or the toxin you are looking at or studying, how toxic is it? So usually you call it lethal dose 50 or lethal dose 100 or lethal dose 75. So that means 50% of animal going to die with that dose, right? So you do a different dosing. And so that's kind of like it's like a radioactive decay time, right? We call it half-life, right? Because you don't worry about how many years does it take to completely, decay the isotope or radioactive molecules, completely disappear, right? We always calculate by half-life, right? Say if it may be 14 years for, carbon C14, right? Carbon isotope or radioactive molecule. And so that's how also for lethal dose, we try to calculate or report based on the 50% or 75% animals. So that's one way you do this. And I think I have a plot for that. So this is how when you do the assay, whether you use any kind of animals, whether it's a C. elegans, the parasite, or you use the larvae or rodent model, and you can always do this type of plotting, okay? And so these are special called Kaplan-Meier survival curves. This is always used. So if you look at the different dosing, higher doses, the mortality would be higher, right? This is higher mortality, right? If you have a 10 to the power 10, 5 to the 10, and you can see the mortality is much higher, as the dose decreases, you can see the mortality decreases. And so this is called survival probability or percentage, got it? So this is the statistical program, we always use that. This is for some other ones. So these are some of the examples how we report the toxicity of or pathogenicity of a given organism or molecule or chemical. Everybody clear? You have to do it at some point, remember. Make sure you understand this, even if you haven't done it now. So this is cell model, I mentioned that. If you have a healthy cells, this is vero cell, right? These are like a elongated fibroblast-like cell, and if it is exposed to E. coli toxin, which is the cigar toxin, or sometimes we call it virotoxin, look at it, after like somewhere 48 to 72 hours, this will look like this. So you can look under microscope, right? And this exposed to this toxin and all these cells, do you think they are alive? Looks like they are all dead, right? So this is called CPE, cytopathic effect. Cytopathic effect. This is a terminology we use that. So this normal healthy cells, convert it into more round cells, they flocculate, they come to the surface, they detaches from the plastic. So these cells, epithelial cells, they always attach to the plastic. That's how they grow. And then when they're dead, they can come off. They become granulated, they become easy to observe. So sometimes you don't need anything, just look under microscope, you'll know it. Sometimes my eyes also, sometimes if you are well trained, you don't need even a microscope. I can tell you just looking at this, I can tell all this certain percentage of cells dead. You know that it's toxic. Okay. So this is how we do it. Otherwise, you can stain them, stain the cells. It's called live death staining. One is the common one, it's called trypan blue. And so if you look at these clear cells, if you add the dye, this dye is very easy procedure. And if the cells are alive, they don't take the dye. If it is dead, the dye gets in. So this is live cell, this is dead cell. And if you have seen hemocytometer, the one we use it for blood cell counting, and you load it there, then you can count it under microscope. Then you get a live dead estimate. And this is an essential tool for immunological assays. They always do that. Or anytime you are doing cell culture, you have to know how to use hemocytometer to count the cells. These are not bacterial cultures, so you have to grow them on the plate, count it, not like that. This is like you have to put them in this counting chamber, hemocytometer, and you count this. Okay, you are either counting live cells, that's the best way to do, because a lot of cells would be dead, or you can count just the dead cells. Either way, you can calculate it. And then at the end, you can calculate the percent dead, right? It's pretty easy. Total number of cells you used, right, divided by the number of cells already live or dead times 100, right? So that's very, very easy. And you have to know the calculation, how many cells. If you find 10 cells and it's like a 10 times 10 to the power 4, that's the calculation. It's very simple. And you don't need a calculator or anything. Just you give me the number, I will tell you this is the percent cytotoxicity. You can easily calculate it. So these are some of those other graphicals or pictures. So you can see this is control. If you add vero cells, if you stain it with the trypan blue, you can see this lot of dead cells. This is another control, maybe non-pathogenic E. coli. It's not killing it. These are some of the lymphocyte cells also respond to the toxins. This is already I mentioned that, right? These are all dead cells. These are still alive. These are lymphocyte cells. So you can also use electron microscopy to assess the cell damage. It's very simple and look nice. So this is B cell. B cell harvested from animals. I think this one, or I say human B cell. And when you look at the B cell from humans, they got a lot of the surface molecules. And if they are sensitive to toxins, a lot of those makes a big hole, and it loses all the small molecules. This is also more uninfected versus these are infected with Listeria monocytogenes. You can clearly see the damage. Other way, what is the other best way you can do? Those are like a microscopic or observational, but in this case also this is the most easy, and I like this. So you are measuring enzyme release, right? If the cells are damaged, the membranes are damaged, they will release their internal enzymes, right? And if you have a proper substrate, you can detect that enzyme. And the degree or level of enzyme activity corresponds or correlates to the amount of toxin you have or pathogens you have. Okay? So this is, you can use it called alkaline phosphatase. It's one of the enzymes many cells has. And then if you add the substrate, they will change the color. So if you, these are some of the substrates. So paranitrophenyl phosphate. Nitrophenyl phosphate is the substrate for alkaline phosphatase, right? And then when the phosphate is clipped off, the phosphate, the benzene ring, that's yellow color. Okay, that gives you the color. This is how you assay it. And these are really powerful tools. You can generate so much data for doing this assay. Sometimes you can also add proper substrate, so methyl amblyferol phosphate, and it can give you fluorescence, which have a higher sensitivity. Okay. The most commonly used lactate dehydrogenase assay, or called LDH assay. So the cells carry lactate dehydrogenase, right? If the membrane damage, the enzyme is going to get out, and you can add the substrate. And this pathway actually it converts tetrazoleum, which is yellow, to formazone, red. And this the pathway it goes, lactate dehydrogenase convert lactate to pyruvate, and the NAD converted to NADH to NAD, and during that time this hydrogen ion obviously contribute to these changes in the substrate color, okay, and that color you can measure it, and that's how you know that LDH is released, like the alkaline phosphatase. People also have used the beta galactosidase. A lot of these enzymes assay can be used to assay for the actual action. There is also other assays called MTT assay. These are more metabolic activity. Other is called WST assay. These are also again determining that metabolic activity of the cells. Also you can do if the cells are going through apoptosis. You heard about apoptosis? All of you should know, right? It's a programmed cell death, and opposite is the necrosis, right? So one is called homicide, other is called suicide. Necrosis is a suicide, right? Killing themselves. And the other way around. Homicide is the necrosis, and the suicide is the apoptosis, programmed cell death. Do you know why cells do that? Do you know why they undergo apoptosis? You have some idea, right? Yes, some of you have some ideas. So we notice that many diseases, when there is pathogens or toxins coming into the body, so the cells try to kill themselves, right? Why? Because they don't want that gene to be propagated, right? So the apoptotic cells do not induce inflammation. What it means is that the macrophage will clear up those dead cells. So the genetic material is no longer has the ability to transfer to other cell, okay? And that's a different strategy. Whereas the necrosis, if the cells die, the gene or DNA remain intact. There's a possibility that other cells pick up the genetic material and then they will perpetuate that problem, okay? So a lot of viral infection or bacterial infections, we have noticed that this is the defense strategy. I don't want the cells when they're being infected by Listeria or Salmonella or some viruses, and they will integrate their genes, right? And we don't want that gene to be perpetuated, propagate in the body. So that's why if I kill myself, okay, and this body, the macrophage is going to come and just swallow it and destroy it, and there is no trace of those genetic material. Necrosis, on the other hand, if it happens, it induces a lot of inflammation. A lot of immune cells come in. Everyone coming in, they will try to clean it up, and in the process, that genetic material can be transferred and can be maintained. Okay. So cancer cells, do they go through apoptosis? No, right? So if you can find a way to induce apoptosis in cancer, then you got a cure, right? Unfortunately, those are you cannot kill them, right? They metastasize eventually, and so you cannot kill them, and that's the problem. They don't go through because they have the proper gene to maintain that status, right? So apoptosis, you can study this because this is a tool, right? Now you have to know when and how I'm seeing the cells undergoing apoptosis or necrosis, right? All of those changes. So for that reason, you need to know how do you assay them. Okay, so there is a different way you can do this, stain them. These are different types of staining you can use it. And annexin is one of the really powerful tools, annexin 5 binding assay. If I have more time, I would have explained you a lot of this stuff. But if you look at the cells going through the apoptosis, cell shrinkage, the geiosis stage, I will show you the pictures actually. I think I may have pictures. These are some of this data from our research. And you can see the cells, intact cell, if it is going through apoptosis, you will see the nuclear material eventually degrades, becoming a small fragment, and these fragments getting called bleb, and then eventually macrophages clear them up. These are the standard procedure or step. Okay, that's how this these cells taken care of. And if you stain them by fluorescent or acridine orange, you can see these changes taking place. Okay, all of this. And then eventually it's making this blade. So it's fascinating. And the reason again, what research you will be doing in future or whatever areas you may be. And so you may have to study, understand whether they are going through necrosis or apoptosis. Especially in cancer research, so well, I want to come up with a drug to treat the cancer, then can I induce apoptosis? What signatures you have to look for, right? The DNA degradation is one of the major output. Always DNA get fragmented. So that much I wanted to tell you. But if I'm giving a lecture to my students, I will spend a lot more time to explain every steps of it. Unfortunately, you cannot dedicate that much, but I want to give you a flavor of what are needed or what we understand from this cell-based assay. Okay. We talked about adhesion, right? We talked about attachment quite a bit. How do you do that? That's very simple actually. You grow these cells, add your bacteria, and after some time, you just wash it off, like a 30 minutes to 1 hour. Then you assay how many of them are bound, whether you want to stain it or you want to plate it, right? Then you look for count, colony count. And this is how we know adhesion. If they are gone inside, how do you know they are inside the cell? So what you have to do, in order to monitor the cells inside, you have to kill the cells which are outside first, right? So you have to use antibiotic to kill outside cells, and then after you wash it, then you find out how many inside. You have to lyse the cell and get the bacteria out. And so those processes are very straightforward, and you can do this. These are pretty simple. You can again follow this, and that way you will know. And this is the cell, actually a neuron, nerve cell I mentioned, keep on mentioning. These are nerve cells bound by Listeria. Anyway, and the other one you can see is that if the cells are passing through to cell layer, right, paracellular. And I showed you this picture before. This is also a very powerful tool for those who are studying gut health or gut permeability. This is what we do. You have a transwell insert. There is a filter membrane underneath, and you grow your cells. So for example, you want to grow KCO2 cells, you want to grow HT29, or any of those intestinal cell lines, or even other cell types. You grow them in a monolayer, and when they are growing in the monolayer, and you can measure their electrical impedance. We call it TER, translocated epithelial electrical resistance. What it means is that, how do you know they are forming the monolayer? It's very difficult to see them under microscope. That's why we have a probe. We measure that electricity. You all had physics, right? All of you, undergraduate, I mean high school level physics at least, not the college level. So how the electricity move? Negative to positive, right? And if there is a water in between, you have two electrodes, right? It will flow one type of resistance, right? Always resistance, right? And that's called impedance. Impedance means impede the progression. That's what we call impedance. Okay? Conductance means, I'm not an engineer, so I'm trying to teach you a little bit. So the electrical conductivity we are trying to measure. So the water will have one type of conductivity, right? If you add salt, you will have a higher conductivity, right? So the ions which are transmitting the electricity. And so here, if you put a probe here in this one and one outside, and then the electricity movement would tell you the integrity of the membrane. If these epithelial cells made intact monolayer, there will be high or low resistance. Low resistance, right? So that means electricity has opposite. This is where I get, it's not confusion. So if you have an intact cell monolayer, you have a high impedance, means high resistance. So we measure that, say, get close to 500 ohm, right? And that's the resistance. But if the cell monolayer is damaged, the resistance would decrease, right? Because these cells are not intact, and therefore electric current movement would be faster or quicker, because this liquid is actually ionic molecule, right? A lot of charge there. And so based on that, we determine the integrity of this membrane layer. Okay? And so that's the thing we do, called TER measurement. So again, if you read it up, if you in future you need to use this type of tool, you'll find it interesting. And other thing you can do also, if you add bacteria, if they go through, and they will be large number here. If somehow blocked, then you will not see them downstream. Other thing also, you can use also dye, put some kind of fluorescent dye on the top and see after 1 hour or 24 hours later how much of the dye at the bottom layer, and that tells you the integrity of this membrane. The reason I'm spending a little time on this, because a lot of those diseases, even this heat, have you ever suffered from like a vomiting after coming from heat or diarrheal episode? You had any of you? It's a common practice. I think 2019, I came to India, and I went to Varanasi, right? And that was happened to be summer. And I went to one of those parks to visit that area. And that day, temperature was close to like what we were experiencing. That day, I think it's close to 42 or 43, if not 45. It was a very hot day. And so we end up, I think we are kind of naive, not really paying attention to the heat. It was kind of like a dry heat. And so we spent like an hour or 2 hours going around, but we had umbrella and stuff. And then immediately in the afternoon, we started showing symptoms of diarrhea, okay? And especially me, and then I think one of my children also showed the symptom, but no temperature, nothing else. And we know that it's not coming from food, because none of the food we didn't eat outside, everything is well prepared. And the reason I'm telling you that this is the first time I experienced it. I never had that type of episode. And then realized we suffered for like almost 2 days, continuous, and we're trying to replenish with electrolyte, always have a lot of electrolyte, right? And then you study more about other people, those who are long distance runner. This also sometimes, you notice that when they're running marathon, sometimes they throw up, right? And also some people suffer from diarrhea. And these are non-infectious diarrhea, okay? So what happens, some of those stressful conditions, it makes our gut called leaky gut, okay? This epithelial lining in the gut, it behaves like it's infected by, say, cholera organism or Shigella or Salmonella. Shigella is different. So you lose a lot of fluid. So what it does actually, it opens up your gut barrier. So it makes it leaky gut. And this heat stress makes it much leakier. Heat, some of the stressful conditions, sometimes you know that people when they get really stressed or nervous or anxiety, they vomit, right? Notice that? It happens, right? A lot of those natural response. What happens, you can define it by saying that your gut is leaky, so all those fluid coming out, okay? And you can study that, on this type of model to show that the leaky gut can happen. And I'll show you a little bit more data. But you can, in the laboratory setup, you can create that. So this cell, if you're looking at these two cells, this kind of separates out. I think I showed you some pictures, I will show you a little bit more. And leaky gut is common, is the main reason for a lot of diseases we suffer from. Name any disease, see if you can find a link with the leaky gut. Parkinson's disease, Alzheimer's disease, those are the serious diseases, right? Inflammatory bowel disease, all of those, heart disease. A lot of diseases are now they're finding that it's attributed to leaky gut. Why? Because of the microbial community we have, right? What microbes has? LPS, peptidoglycan, all those nasty surface molecules. So all of those microbial, billions of bacteria or microbes, their molecules being absorbed to our system. Immune system responds badly. And that's where we are finding out the leaky gut is responsible for many diseases we suffer from. Even to maintain your own health, think about it. So I wanted to give you the science behind it. And so the leaky gut mechanism we are finding out is responsible for so many diseases. I have a huge list of those. I don't know if I have that slide or not, but I will show you those list of diseases come from leaky gut. And we usually study using this. Anyway, so these are the cell-based assays you can do it. And also you heard about the whole body scanning, right? Bioluminescence. This is the whole body bioluminescence imaging. And if we have a bioluminescent bacteria, you feed the animal, as the animal move from organ to organ or different places, and you can take a real-time picture. These animals are not actually dead, these are live animals. And you just anesthetize them for a certain period and take an image, and then you can bring them back. So that's why we call it live animal imaging. Like the human body, we do MRI and all of those live scanning, and these are actually bioluminescence. We put the marker to measure that. These are more studies done. So let me go back. One thing I wanted to highlight. How do you know you have a leaky gut? Usually, if you go to a gastroenterologist, if you have a intestinal problem, they will give you some kind of dye. And you will drink that dye, and after 1 hour or two, you can collect urine and look at the dye level in the urine, okay, or in the blood. And if you have a high level of that dye in the blood or the urine, that means you got a major problem in your gut. So that means you have a leaky gut. And that's where how to treat your gut so that you can close them. Not everything needs to be getting into our blood circulation. So we do that, follow the dye marker, okay, just to give you the idea how we test for leaky gut. Anyway, I know it's already 1 hour. Any questions on this? Go ahead, Rohan. Use the microphone.
Sir, you mentioned the relation between Parkinson disease and leaky gut. Can you explain and give more insight to this? Because Parkinson is necessarily a neurological problem. It's a critical illness being discovered.
So again, I have to go back to the microbial components. Okay, they are taken up by the gastrointestinal tract. Eventually, get to the molecules. All those molecules get to the body, right? The blood circulation. Then you don't know exactly how the immune cells responding to each of those molecules. And so, if it is a neurological disorder, some of those molecules getting to the brain, and the brain also getting damaged. And same way with the Alzheimer's disease, it's forming this plaque. A lot of those plaques actually derived from these microbial components are playing a major role. Okay, so varieties of chronic diseases, including heart attack or heart disease, also the stroke. People suffer from stroke, right? And forming this small clot in the blood vessels. And these are all we're finding out is the microbial origin. Because of the leaky gut plays a huge role. Besides the cholesterol or other molecules creating a plaque. So it's a huge. That's the current trend of research. That's the modern approach to finding out the causation or causative agents for, including arthritis. I think we mentioned arthritis other day. A lot of those molecules also antigen-antibody interaction, and they're complex forming, and they're traveling throughout the body, and how immune system responding. This is a complex process, though. I'm trying to simplify it. Okay, but these are all linkages they're establishing. So I think it's a kind of an alarm, but I think same time it's better to know that because there are some recent review articles, they cover all of those things. If you want, I can bring those papers or send you the links so that you will study this, how a lot of those leaky gut, especially. The word leaky gut, as soon as you hear it, so you should be alarmed, am I keeping my gut healthy? So that's why I focus on gut health. How do you keep it healthy? How do you prevent the leakiness? Right? So I'll try to tell you some of the solutions. So that's why we take right nutrient, proper diet, right? And a lot of these toxic molecules, they cause this barrier to open up. I've been studying this leaky gut for a while. So the proteins, I showed you some pictures, right? How these proteins holding the cell structure together, and it's fascinating. The microbes manipulate it, or different conditions can manipulate this signaling cascade and open up the junction. And some organisms just wipe out that totally. So from infectious disease point, it's very important. Also from the chronic diseases people suffer from, or the type of diet you eat, that can also influence. A lot of factors can influence the gut integrity. Okay, and so it's not just one factor causing it, but the leaky gut is really an important concern. Always you should be thinking of how can I keep my gut healthy? Think about that. Healthy gut. Okay, I know it's time for break, right? It's 10 to 11. I think 5 past, so we'll start the next lecture in 1 hour, correct? I forgot. Let me check my.
Sir,
Yeah.
I have a question.
Yes, go ahead.
Sir, my question is also related to leaky gut.
Yeah.
Like, so you explained that when we have a leaky gut, then we can detect it by using a dye and stuff.
Yeah.
But usually, the persons with who are suffering from leaky gut suffers also from frequent diarrhea, even though their diet is optimal, they are having optimum nutrition,
Yeah. Yeah.
and still they suffer from diarrhea. And when they visit a doctor, the doctor usually recommends them having a fiber-rich diet.
Forget what?
Fiber-rich diet, like they add fibers in their diet, like they have a salad or they can eat fruits, so that they can have a good amount of fiber in their gut.
Oh, fiber rich.
Yes,
Okay.
fiber-rich
Okay.
diet.
Okay.
So, sir, my question is, how does a fiber-rich diet affects a healthy gut microbiome? Like, what exactly fiber does to our gut?
That's a very good question. Yes, depending on what type of problem you have, I think fiber could work against it, or fiber could work favor. Okay, let's talk about the favor. What happens is the type of microbes or microbiome you need, they like the fiber. Fiber is very healthy, right? Everybody talks about eat fiber rich food, right? You have to select the food that is rich in fibers. And when you say about fibers, means those are complex carbohydrates, right? It's not easy to digest, right? So that means there is a group of microorganisms, they can break down those fibers and then harvest that energy from that. One of the byproduct of that is that this metabolism or fermentation is that producing short chain fatty acids. One of these short chain fatty acids, you heard about the butyric acid, acetic acid, lactic acid, propionate acid, right? These are the short small molecules. A lot of times they have antimicrobial effect, but especially they are finding out the butyric acid is really good for your health. The way it works is that it's getting absorbed into the system, and then the epithelial cells like the butyric acid. You know, the butyric acid helps their repair. Also, the immune cells respond positively to butyrate or butyric acid. And so that's one way. The other thing it does, the type of microbes which like the fiber, they're enriched now, right? So if you have a different types of, if you are eating a lot of fatty food, a lot of protein rich food, so you will have a different group of microbes, right? If your diet rich in a lot of carbohydrate, polyphenols, a lot of those healthy stuff, right? If you are eating a lot of blueberry, berries, you know, blackberry, strawberry, all kinds of berries, they are also rich in polyphenols. And so together with fiber, which acts as a prebiotic for like probiotic organisms, and then polyphenols, a lot of these are going to make this microbial community more towards the healthy. And one of the healthy group would be the probiotic. We already have our probiotic organisms. And if you enrich their number, and the probiotic organism, I will show you a little bit more data, is that in general, their role is to make the tight junction tighter. Okay, they just work just the opposite. I will show you some of this data research next week, that how if you have a leaky gut and if you start eating this probiotic, and they will make it tighter. And so those are some of those reasons it works. Make sense?
Yes, sir. Thank you.
Any other questions? Okay, so I know we didn't talk about biofilm today, right? So this probiotics, when you give them this food, fiber-rich food or any of those food, you are enriching these probiotic organisms, right? And how do you keep them? If they can form biofilm on the mucus layer, that means their persistence is going to be longer. Can you make this probiotic a biofilm-rich environment so they will be maintained and they can do this positive work? We started doing some work. That's the positive application of biofilm. If you can make these probiotic organisms form biofilm, they will do their job. They will do their magic. Okay, so you can always find the linkage. I didn't talk about biofilm today, but this is how you can see the positive effect of biofilm in helping with the gut health. Good. Okay, I know. Take a break. We're getting close, right? We're going to have enough short. Raise your hand. Any immunology you had? Okay, very few people. Okay. So immunology, when you take, it's like a full course, like a 1 semester or 2 semester long. It's a long subject. And then sometimes also if you take microbiology or medical microbiology type, then there are some like a small immunology section, right? Not the complete, but a little bit, right? Any of you had that level of exposure? Like a little bit of immunology. Okay, some of you had a little bit of immunology. Next 2-3 lectures I'll try to talk about immunology, right? Essentially the way I look at it is that, you heard about that if there is, so if a tree falls because there is a wind or storm coming, right? If a tree falls in a forest, how do you know the tree fell? It's kind of a stupid question, right? So what I'm trying to tell you is that if there is a storm and there is a tree in the forest, it fell, right? Follow me. And you are here, you are not in the forest, right? You are here. How do you know a tree fell? You don't know, right? It may fall, right? It may not fall. But if it fell, right? You still don't know, right? It fell. Okay. So that's one thing. So let's see if you encounter, okay, that's the unknown, right? So now let's talk about your health. Okay, it's like similar thing. So if there is some microbes get into your body, right? And the question is, how do you know your immune system responding or immune system is inactive? A lot of times you will not know, right? Until there is outcome, certain outcome, right? So let's say if you have a pathogen, 2 of you are sitting there, then I gave you a culture and both of you had a chance to drink that or swallow it, and one of you became sick, one of you did not, right? So what does it tell you your immune system about this 2%? Because one of you became sick, right? Other did not. If you take all those factors, there are so many factors can happen. First of all, I gave you pathogens, right? And the point I'm trying to make is that one person became sick, some kind of showing symptoms, right? Another person did not show any kind of symptom. This is pretty natural, right? In a community, whenever you expose, some show symptoms, some don't show symptoms, right? So now I will ask you, what do you think the status of the immune system in those 2 persons? Right? The person who became sick, what do you think that immune system looked like? Weak, did not do its job, right? Right? And the person who is still healthy looks like he defended very well, right? So that means that immune system worked for that person. So that's how I'm trying to interpret that defense, host defense. Okay. So if you think about the immune system, always to me, it's a battle or fight between 2 entities, right? One is the pathogen and one is the immune system, right? So when there is a battle, always I feel there is a winner and a loser, right? Or there is also draw, right? 3 outcome: winner, loser, draw, right? Okay, so probably by now, if I say somebody won, that means if the bacteria won, your immune system lost, right? So you equate that to infection, correct? If the immune system won, bacteria lost, that's when you said you are not sick, right? That's the 2 outcome. Then there is in between, it's like who are those in between? That's the no winner, right? Or both won, or they are same, right? What is that usually look like? Huh? Chronic disease, it happens. Chronic cases can happen. That's like a continuous fighting. That can happen. But if you take the acute disease, right, acute outcome, the death or alive, or real sick versus not sick. So there is some in between. Sometimes you feel bad, but not full blown sick. Have you seen that? That's called subclinical infection, right? So there is a fight happened, but both the bacteria, both the immune system, they fought because there is expenditure to that, right? Because it's not that bacteria lost something, immune system lost something, and during that time that also can show some symptom, or we call it milder symptom, like a subclinical. Okay, so all of these are actually dictated by how your immune system or host defense respond to that pathogen. That gives you some general output, something to think of. So immune system, to me, I just look at them. What is the outcome? This is outcome basis, right? And then who are those players going to come and interpret, or this type of interaction, play important role in this type of interaction? That's what you have to think, right? So keep this in mind: winner, loser, and then in between. And then, of course, if there is a continuous fight forever, there is a certain duration, right? When you become sick, you usually recover, depending on the type of disease, within short period of time, right? So that's the immediate response from the immune system. But in some cases, like he mentioned about the chronic diseases, there is a no winner, no loser, and it just keep on going forever. There are some countries used to fight for many years, and that's the chronic diseases, right? So essentially, when you look at the immune system, it's actually battle between immune system and then the pathogen, right? And so always you're trying to figure out who are playing role and how can you change that direction to make somebody winner, right? Of course, you don't want the microbes to win, right? We want the immune system to win all the time. So what can you do, or how do you understand that to make immune system is the winner so that you can defend better against the pathogen, right? That's our goal all the time. Okay, so with that background, let's talk about immunology a little bit. And I know what I'll do, because it's really a lot of component of the immune system to understand how every player play a role to defend you against some of those infectious diseases. So therefore, it takes a lot of time. But I'll try to cover within 2 to 3 lectures to give you overall general information regarding how immune system works. Okay, and then of course we'll think about the biofilm, how your immune system play a role against a biofilm. Okay, so this is the introduction I will use. It's a host pathogen interaction, right? Host is us, pathogen is interaction, right? And so there's a subject, we call it host pathogen interaction. Essentially, the interaction with the immune system. So whenever you try to understand a disease, you always have to understand how immune system works. Okay, that's the immunology is so important, because you cannot explain a disease without studying a defense system, right? So that was the first question I asked you. If a tree falls in the tree, how do you know it fell, right? So the thing is that in order to understand, you need to tell me the tree fell because I was there, right, to witness it. And so the immune system, if it is immune system is responding or observing the tree fell, right, then you can accurately say your immune system is playing a role in that decision making process. Okay, and that's truly host pathogen interaction. And so a disease, just studying microorganism, you cannot really say anything about how it is going to be the outcome, right? Are you following me? So if I give you a test tube culture, and you cannot tell me that it's a pathogenic, right? I can give you just simple lactobacillus, and I can say, oh, there is a pathogen, it's going to kill you or do something, but you cannot make that conclusion without studying with the host, right? Are you seeing me what I'm saying? So understand the disease process, you have to understand the host, means us, whether it's animal, or human system, whatever system may be, and that's the interaction, and that becomes the disease outcome. A lot of times you can study microorganisms, any pathogen, whether Salmonella, Listeria, E. coli, viruses, any of those you can study, but always you have to study in relation to the host, because how our immune system going to respond, that's going to dictate the outcome. Everybody clear on that? And so immune system is very important in order to understand the disease. And when you say host pathogen interaction, we are talking about immune system's interaction towards the pathogen. Okay, so I hope I have been able to convince you the immunology is very important subject, right? So other aspect is that we have a lot of nutrients. So pathogens, when they get in the body, they love to be there, right? And we don't want them, but they need their food. They need to be in the body, and so that's why you will find them. And other aspect also, you have to think of that microbes, they are evolving all the time. Okay, the immune system, right? We have immune system, set of immune systems. We can improve our immune system. We'll talk about that. Then immune system try to destroy the microbes, right? But at the same time, if you destroy the microbes all the time, then we're never going to see any kind of disease, correct? But we all suffer from different types of diseases, right? You agree? So then what is happening? Why microbes winning in some cases, or even within two individuals, there is going to be a difference, right? So one aspect I want to remind you, the microbes are continuously evolving, right? That evolution is part of their life. Mutation is part of their life. Now you know that in the news now they are saying that COVID, another variant is coming out. So that's part of the evolution. You have to recognize the microbes are continuously evolving. If they are not evolving, then we will not have this problem. We can solve this problem easily. So the microbes were here before even the Jurassic era, right? Before even dinosaurs, right? Which came first in this living system, in the earth? What microbes was here first? Huh? Archaea? Which one? The sound, the noise is pretty deep. Huh? Archaea, we call it. Archaebacteria. Somebody said virus. Okay. Planktons. Okay. Okay. Let's go back. This is a good argument. I'm sure always you get into this argument. So if you say what molecule first came to this earth, the origin of the life. Okay, let's go back. Amino acids. Okay, anybody else? RNA. Anybody else? Which came first, egg or chicken? See, that's where we have a controversy. Which came first, chicken or egg or chicken? Always think about this, the beginning of the life. This is a question I ask my students also, what is the first molecule arrived to this life, where the life began. So somebody said RNA. Anybody said DNA? You started with the virus, right? Amino acids. So amino acid can replicate. See, we're getting towards his block. RNA. Not DNA, right? How about protein? Protein cannot replicate, right? Okay. Actually, the argument is actually RNA. Okay, because that's the simplest molecule when they assemble, and that's the beginning. Is even single-stranded RNA can withstand, right? That's the beginning of it. So read up a little bit more about this, because this is fascinating how everything started, and from there the microorganisms evolved, right? And then of course the amino acids you cannot replicate it, right? Yeah, amino acid could be there, but how good it is going to be in terms of the yeah, but I'm thinking from the standpoint of is it going to replicate, is the starting of the life. So it has to be nucleic acid, right? And the RNA is the logical because it's single stranded, and if it is a virus particle, and that can replicate within a host, any kind of living organism, and that's most scientists believe that RNA is the origin of life. Okay. Anyway, so getting to this point is that the evolution happens, right? The mutation happens, okay, and the organism always evolving. And the RNA, the also other thing is that RNA viruses are very what? Their proofreading ability is very low, and so there is a high chances of mutation in the viruses, and that's the reason virus is very difficult to control, right? They are always mutating, and because they don't have any proofreading enzyme, good polymerase enzyme, and that's why we see the more error in their DNA, the nucleic acid synthesis, and that's why every generation you may see some kind of mutation. So I'm not going to be surprised the virus's new variant going to come most of the time. Bacteria, on the other hand, you don't see that. This is more stable nucleic acid. There is a mutation, there is a evolution, there is a changes, but it's more like acquisition of genes, right? And that's how they get a lot of different types of mutation. Okay, but virus just they are prone to mutation very because of the way they replicate. Okay, so that these organisms are always going to change. So if your immune system designed for one type of virus, one variant, and then next time that virus came as a little bit different, then your immune system kind of go out of, cannot recognize anymore, and that's when it fails, and that's why the disease happens. So this system is called dynamic system, always happening. So how do you alert your immune system to help protect you against that viral or microbial interaction or infection? So anyway, commensal and opportunistic pathogen you have to pay attention to. They are naturally there, they are not causing disease, but when your immune system is weak, boy, that's when they get you. Normally they will not affect you, but that's when it happens. But that's aspect you have to remember that, because your immune system is weak, right? Most of the time when you know that you are stressed or you are not feeling well after a long journey, everything is kind of like a downwards, and that's when you see sore throat, right? Or coughing, or flu virus shows up, or mild fever, all kinds of symptoms shows up. And a lot of times it's because your commensal organisms, say Staph aureus or Streptococcus, right? They started to flare up because they realize that your immune system is not protecting or robust all the time, and for that reason they start to showing weakness, and that's when the disease happens. So as I said, immune system is battle between pathogen and host, and one wins, one loses. So depending on who wins, that will be the outcome, right? It's pretty obvious. So anytime I become sick, then I think that, well, my immune system is not doing its job, right? So personally, you can do also a lot of things. How do I keep my immune system active, right? Those are the different aspects, whether it's athletics or good diet or good habit. A lot of those things can make immune system healthier so that they can respond to the invading pathogens much better. That's the way I translate it, right? So think that way. So immune system positively. So now let's drive through it very quick. Two types of immunity, we call it innate and adaptive. And innate means what is there in your body, whatever you are. Okay, your entire system in place. That's the natural system, natural immunity we call it, or innate immunity. So innate immunity is generally very constant. They don't change much, but that thinking is changing now. A little bit saying that, can I train my immune system, the innate immunity, called trained innate immunity. It's not adaptive. Adaptive is like you are exposed to certain antigen, your body responds to over a long period of time. That's called adaptive, okay? And it takes longer, but innate system is there always. If pathogen comes in and your innate system is trying to protect you. That's the first line of defense. Like if you think there is a two door, first door is the innate system and second door is the adaptive system. Okay, if innate fail, then they get in. Then if the adaptive fail, then you show the symptom, right? And so this is the way it works. So I will give you some examples. And innate system is natural, non-specific, cannot be induced. But that's wrong, I think, or it's debatable. This cannot be induced, it's changing. Okay, we'll talk about that. And then adaptive system is induced, it's highly specific for a pathogen, requires longer, 4 to 7 days for response, slow to develop. Usually you don't get a adaptive response right away. It takes a while, right? So you went to a restaurant and you ate some food, giving an example, quick example, and then within next day, tomorrow, then you started showing, oh boy, I have a severe stomach cramp, right? And I'm started to throw vomiting. And then you are traveling to bathroom 5, 6, 7, 8 times. 2 days later, you've been doing this for a couple of days. This going back and forth to the bathroom, you are miserable, you are sleeping on the bed, and then third day or fourth day you woke up, you are feeling much better, right? I say, oh, it looks like it's gone. Okay? Tell me what happened to your immune system knowing what you just learned from my story. So first thing you can say, your innate system didn't work, right? Okay. What about your adaptive system? Probably didn't do anything, right? Because it takes longer, it's not there. So I'm giving you an example. Certain diseases, you just have only innate system to work with, nothing else. Okay? But some diseases, yes, you have adaptive system. If you are suffering from Listeria infection, which takes 2 weeks, 3 weeks, or mycobacterium infection, or some other chronic diseases, or the diseases which show symptoms many days later, most likely your adaptive system is playing a role. So depending on some diseases, think about this. The showing symptom right away within 30 minutes, 1 hour, 2 hours, or 1 day or 2 days, your adaptive system hasn't done anything or not doing anything. Got it? Okay. So let's look at the innate system, adaptive system a little bit more. Well, you probably know historical perspective about the immune system, how we learned it. Edward Jenner is the father, we call it, you know. His experiment was actually the pox virus, and that's how the vaccine terminology came. The people who are the milkmaid, we call it goala, I think in Bengali, those people who milk, right? And they are always in contact with the cows, and the cowpox, they may have exposed to cowpox, and those people never got the smallpox. Okay, I know smallpox is now eradicated, but with time to time we see chicken pox, we call it. This is the milder form of the disease. So the smallpox, so that's how we actually originally the terminology vaccine came. Vaccine came from cow, vaca. Vaca in Spanish called cow, right? And so this experiment, this cow experiment, so what he did actually, he took the cowpox pustule, like this thing, this type of pustule, and he removed that and he injected to 80-year-old boy who never developed smallpox because he already got vaccinated with this cowpox vaccine. And so when this boy was protected, then he gave this term as they call vaccine, and vaccine term came from vaca, means cow, and so that's how we use the word vaccine. So definitely he is the father of the vaccine, or our understanding of immunology. And of course Louis Pasteur later on developed vaccine against anthrax and rabies. And so these are showing actually how the smallpox look like. You may have seen these pictures before. Luckily this been eradicated and we don't show that. But the one good thing about smallpox is that if you have once chickenpox or smallpox, and you have a lifelong immunity, you never have to take a vaccine. Or if you had taken vaccine, one time against smallpox, you are protected entire life. But so bad is that not all diseases are like this, right? So, okay, we'll examine that why. Okay, very simple, again, quick difference between innate system and adaptive system. So, the specificity we mentioned, they are not specific, less specific. Adaptive has a high specificity. Diversity we'll talk a little bit later on, little bit limited but very large. Memory response. When we say that lifelong protection or long protection is because of the memory. Okay, we'll talk about that. And then non-reactive to self antigens. Probably they are not, none of them. Different types of molecules are responsible. Proteins, complement, these are TLR, antimicrobial peptides, these are antibodies. Like everything in the body naturally present, and including acid, mucus, all of these are part of natural immunity. Okay, and anything immunologically speaking, the cells, the antibodies, the cytokines, these are part of the adaptive response. And action for innate, this is immediate, right? Immediately it's providing protection. If that fails, and then you see the disease. So for example, acid, let's take stomach acid, hydrochloric acid, right? We all have that, right? We discussed that. If HCl is there, and then if you have a pathogen going through your stomach, then HCl is supposed to kill it, right? Destroy it. So that's an immediate action. If it kills it, then you don't have any disease, right? But if it fails to kill all the organisms, then you have the disease, right? And that's essentially what happens when you see symptoms within short window of time, before the adaptive response wake up and do that. And so this is a quick, clear example that the innate system is immediate action. Clear? Okay, and if you look at the adaptive, we're going to break it down, few of these innate, adaptive, and then at the end we're going to talk about immune response against biofilm. And so whole idea is that I'm not going to make you a master of immunology or learn everything about immunology, but I'm trying to tell you in a story, in a way that you have at least some knowledge from the immune system, and how immune system, at least think from your side, how immune system protects me against different diseases, how my immune system is going to help me to defend against some infection, or how a vaccine works. So that's my goal within next 2-3 lectures. Okay, so let's talk about innate immunity. Like I said, it's a first line of defense, right? It's the first thing the microorganism going to encounter. Naturally present, cannot be induced, I said, gastrointestinal tract enteric, multifaceted. Example would be skin, epithelial barrier, mucus, mucous membrane, a lot of complement proteins, antimicrobial peptides, acids, resident microflora also part of innate immunity. Toll-like receptor, we'll talk about that. Some molecules also called PAMP, it's called pattern recognition molecule, and these are some of those part of toll-like receptor. So just to give you an ABC of GI tract or the human body, I'm sure you read in your biology, right, how and what we have. Let's review it, what we have. That way it will help you to understand a little bit better. So if you look at the human body, I will focus a little bit on immunology first, but if you look at the gastrointestinal tract, the esophagus, then the stomach, and then of course that leads to the small intestine, and the small intestine has duodenum, jejunum, ileum. Large intestine has a cecum, it's called ascending, then descending column, right, and colon, and then the rectum. And then this is liver, this is spleen. These are very important organs, right, tissues. And you got the bone marrow, the long bones, and so there is a bone marrow, right. And so from the immunology side, immunological response, right, so we are thinking that whole body. So one way microorganisms get through our body through mouth, right, then also they get through respiratory tract. Also they can get through the skin if there is a cut. Skin provides good protection. So depending on where the pathogen is coming, locally you need some response immediately, right. So the lymph node, these are very good immune system where immune cells are sitting there, right? And so if some happens, the respiratory tract or oral route, pathogens get through, so your local immune system, which is the lymph node, they are going to respond, right? So when you go to see a doctor, first thing what he does, he check your lymph node, right? Underneath the ear or submandibular lymph nodes. So what happens, if your immune system responding, they will enlarge. Okay, that's the reason they are checking it. So they are saying that infectious agents are already coming into the body. They are going to check your lymph nodes. That means, is your immune system responding well to the infection? That's a good sign whether there is an infectious agent involved in this whole process. Okay, and so the lymph nodes we have throughout the body, large number of lymph nodes. Same way we have lymph nodes in the gut, which is called mesenteric lymph node. If it is a gastrointestinal diseases, the mesenteric lymph node is going to respond. This is going to enlarge. And then we have all those lymph nodes throughout the body. Okay, so these are the primary lymphatic system or lymphatic tissues or lymph node or immune system that play a role. And thymus is another one. We have it right here. Thymus. Thymus is actually the first primary immune system, we call it. A lot of immune cells originate from thymus. One of the main one is called T lymphocytes. That's why it's called thymocytes. And they start here. But as you grow older, thymus disappears. Okay? But still, those T cells are playing important role in defending against infection. So nonetheless, these are the places we see that. So let's quickly overview about the natural or innate immunity. So we got skin, mucous membrane. Let's see if you can all recognize it. Goblet cell, they secrete mucus. Antimicrobial peptides. We have a lot of antimicrobial peptides in the gut. Then toll-like receptors. We'll talk about that a little bit. Resident microflora, and then immune system, immune response in the gastrointestinal tract in general. Skin, I'm not going to go through it. What is the structure of skin, right? We have epidermis, dermis layer, right? Then the blood vessels. Underneath we will see a lot of those immune cells. You can read this. You are going to have these slides, right, with you. And so you can read all of those things. The pH of the skin, the salt level on the skin, right? All of those are going to protect you against infection. Also, we talked about skin microflora, right? So the story I was telling you in that Brazilian, tribal women, and although she lost her community or changed her community, microbial community, but when she went back, she got all this microflora back. It's kind of amazing, right? You lose it within certain environment, but you have the chance to get them back when you are put back into that original environment. That was a long story I didn't tell you, but that's what they found in that women. And they have certain community that keep their skin well preserved against or defended against a lot of skin infection. They have a beautiful skin, okay? And so their microflora providing first line of defense. So we have lysozyme also, right? Skin secretes. We have also phagocytic cells, natural killer cells of the phagocytic cells on the skin, and also keratinocytes. So these are some of the cells keeping our body healthy. So if you look at, this is the structure of the skin, right? If you open a textbook, you will find this. So this is the dead layer, right, which comes up all the time. And if you have a pathogen sitting on that, if you rub it off, the outer layer comes up, it also get rid of all those bad microbes sitting there. You can lose your community too a little bit, but this is what happens. And then if you look at that, this is the hair follicle. And the immune cells, when there is a blood circulation, that's where you will see a lot of immune cells, right? So for example, this is a way to give you a little bit more deeper picture, is that this is the epidermis layer, right? Stratum corneum, this is still epidermis, and this is the dermis layer. You can see this is the dermis, and you can see some of those immune cells sitting there. This is Langerhans cells, and there are more immune cells like dendritic cells, T cells, all of those are sitting there. So one way to understand is that if a pathogen comes in, who is going to grab them first? So this cell, this is Langerhans cells, or sometimes also we call keratinocytes, and the dendritic cells, they are the one going to grab it, right? And then what they are going to do, if they cannot kill it, they will bring it downstairs. They have other cells they can present to them, and then they are the specialized cells, they can kill it. Okay? And so most of the cells here, they could be part of adaptive system, right? All of them on the top, they are part of the innate system, and on the skin, they are more adaptive system. Everybody clear? So even in the normal conditions, this is always happening on your system, no matter where you have the skin. Everybody clear? So this is normally happening. So that's how they are keeping us safe, so you don't get sick all the time. But as soon as you have some kind of breach, right? Have you noticed that when you pull hair, you got some infection, right? All of you can encounter that. Sometimes accidentally or something, you can see the Staph aureus growing, right? And because the organism already got inside, because there is a breach, right? And that's when this immune system try to work, but Staph aureus is going to grow faster on the skin, and then that's how you see the infection. So if you talk about the biofilm on the skin, and these are some of these examples, right? I don't have to explain to you all of these diseases. Atopic dermatitis, eczematous lesions, allergic rhinitis, asthma later in life. So then if you look at the Staph aureus related infection, carbon cold, boil, right? A lot of those, I think if you are unhygienically or summertime, I think this is what very common, because Staph aureus can cause this type of infection, right? Then this is also another Staph aureus mediated infection. It's called SSS, right? What is that movie you had? RRR something? It reminds me that instead of RRR, you call it SSS, right? So it's called, have you seen that movie? All of you seen it? It's kind of funny movie. I never knew that that type of movie exists. Oh, I think my son saw it, and I don't know why he picked it up and said he downloaded somewhere. Then I said, okay, I want to watch it. It's a long movie, but it's kind of fascinating though. But anyway, that's RRR, right? But this is SSS. So it's called Staphylococcal scalded skin syndrome. So Staph aureus, they form the biofilm, and look at the skin, it's almost like a burnt skin. But this is, some people can suffer from this, okay? And these are examples of how Staph aureus forming biofilm and your immune system couldn't do anything. And look at this, what is happening. And then same way with the fungal diseases. You know the ringworm, right? The fungus biofilm form there and continue to grow, and immune system can't do anything about it. First of all, it's happening on the epidermis, dermis layer, right? The immune system barely can reach there. It's kind of dry. That's the problem, right? And then the athlete's foot or the fungus growing on the nail, boy, it's never go away, and the treatment is so bad. You cannot cure them. So these are some of those examples, biofilm happening on this, and how do immune system respond to it? And that's become often a challenge, right? If they're growing, say candida growing on the skin surface, and all of these things happening, but not reaching enough to destroy it. So the immune system against biofilm is always going to be a challenge. If you have a single cell, immune system has a better chance. But if I give you a big blob of things, right, you cannot handle all of those cells together. Immune system can handle one cell at a time. And then we talked about that multi-layer, right? So if the immune cells coming from one layer, it cannot reach the layer on the top. So how do you get rid of it? So always it's going to be a challenge for immune system, no matter what, to control biofilm if it is already formed. You see this? And no matter where they are, on the skin or in the gastrointestinal tract. Physically you can see the picture, right? With their multi-layer, and then the immune system cannot reach. And so that's always going to be a major problem. So we'll talk some of those later on. Let's say what we have in the body, right? We review it. I already mentioned that. Let's say if you are a pathogen, imagine you are a pathogen going through GI tract. What are you going to encounter, right? That's the primary immunity or innate immunity. What do you have in the mouth? Saliva, right? We think we chew food, we have the saliva. What do we have in the saliva? See, a lot of isozyme, enzymes, amylase, a lot of those things supposed to, kill some of those organisms or control it. Then as you pass through the esophagus, you get to the stomach. Stomach has hydrochloric acid, right? We know if it is working properly, it can kill it. But yet we still get sick, right? So what happens is that we don't swallow the bacteria alone, do we? You are not drinking a test tube full of bacteria, right? Always either you are eating or drinking, right? So what happens? Are you maintaining still your pH in the stomach if you are eating with food? Come on, some of you are not thinking. Tell me, your stomach pH is what? Close to? I didn't give you any number, right? Because it's so common, we know that, right? Close to what? 2? Okay, that's a good number. Yeah, from any 1.5 to 2 or 2.5, that's the normal conditions, right? But so that's going to kill any bacteria, microbes going through, right? If they are alone. But usually we eat or drink, right? So microbes with the food. What happens to your pH of the stomach then? Is it still 2? Yes? The question is, when you have the food going through the stomach, that's where the pathogens are, right? Or microbes you are consuming. What is the pH then? Is it still 2? What is your common sense tell you? If you have a little bit of acid, you added a lot of those neutral substance, it's never going to be 2. That's the concept I'm trying to implant in your head. People forget about that whenever we eat microorganisms, we are not just swallowing the microorganisms alone. We are eating with food. Keep that in mind. Food neutralizes acid. So it's no longer going to be 2. It's going to be somewhere pH going to be elevated, right? Acid going to be lower. So going to be close to 4, 4.5 if not. So what happens? Those acid resistant organisms. What if the organism is already in the orange juice? It's already acid resistant, right? Acid tolerant. By the time they pass through your stomach, wow, they are not affected at all. Do you see the reason now why we get sick? So lot of these pathogens, they are getting acid resistant or acid tolerant or acid adapted. That's the point I was trying to make. You have to connect everything I talked in last 3-4 lectures or 5 lectures. Always you have to connect all of those things. Okay, so the stressed organisms, they can survive better in the acid, even though we have a solid acid. Again, also individually, if you have an exam tomorrow, you are studying all night long. Are you going to have a still strong acid in your stomach? No. You didn't know that? When you don't sleep, your stomach gets upset also, right? And that raises the pH of the stomach acid. So it makes you more vulnerable to a lot of these pathogens. Okay? A lot of conditions do not maintain it. What if you are drinking versus eating solid food? Means the pathogens coming through milk and pathogens coming through say dosa or any food, solid food. Are you seeing this? Are you going to see difference? Let me see your thinking. So if it is a drinking, is the organism going to stay there in the stomach long? How long the food stay in the stomach? Anybody knows? Okay, very good. So that's a little long, but depending on what type of food you are eating. So there is a transit time, right? The solid food, 4 or 5 hours, right? They are going to stay, neutralized. They can withstand that. But what about milk? If you are drinking a glass of milk, what is the resident time? How long is it going to stay in stomach? What do you think? Guess. Make a guess. So shorter than solid, right? Yeah, I mean it's shorter transit time. So less mixing with the hydrochloric acid. And so they can go zip through almost if you have any pathogens. So that means it doesn't have to withstand the pH long, right? It can just go through. And so you have to think about if you are consuming a liquid food and you are more vulnerable to that infection. It can be more infective versus solid food. Solid food resident time is longer, they are going to kill it. So same dosing of organism, if I give you with a say solid food and one with the liquid, you will see the different types of outcome. Same dosing. So if you the liquid, there is a chance of getting sick or more, right? Anyway, so you have this natural immunity in place, right? Intestine, you have a bile, antimicrobial peptides, mucus, immunoglobulin, short chain fatty acid I mentioned, we had a little bit discussion. Short chain fatty acid, they are usually antimicrobial, lactic acid, propionic acid, acetic acid, oxygen limiting environment, means anaerobic conditions. Majority of the pathogen we deal with, they are aerobic, and so the anaerobic condition can kill them, limit them, resident microflora and some of those immune cells. So these are the barriers, we consider them as a innate immune system, natural barrier. And if any of those work properly, we will not see the symptom of the disease. But if they fail, that's when we see the disease, right? So what about the physical barrier? So if you look at the structure of the gastrointestinal tract, are you familiar with this? So you have the muscular layer, and then if you look at this is the velar structure inside, this is the lumen, right? We take a cross section. So here what the way the picture is presented, this is esophagus, this is stomach, this is small intestine, and this is large intestine. The whole long several meter tall tube, we try to just compress it. And then you look at these immune cells and this immune system. And if you look at how those velar structure look like, so this is the finger-like structure I was mentioning yesterday. Everybody should be familiar with. And these are the epithelial cells, okay? And this, if you look at this section, cross section, you can see this is individual epithelial cell. And this is what the barrier, epithelial barrier look like. And it's a thin layer, extremely thin layer. And this is called crypt, the bottom of this. And you know any function of the crypt? The crypt, that's where the stem cells are. We talked about stem cells this morning. Stem cells, that's the origin. So what happens, these cells, this is a good thing to understand. They origin right here in this base, the crypt cells. And as they mature, they move upwards. Okay? And the dead cell come off the tip, it falls off. So cells are moving up gradually, replacing the dead cells from the top. So that means each cell has a 10 to 12 days of life cycle, right? So they are dying continuously. They are replaced by the new cells. It's like if you have a cut wound, it repairs pretty quickly, right? So similar strategy. So naturally these are happening, and this is how they fill it up. And this called tight junction. Already I showed you a little bit before. So if this is a Paneth cell, is also another epithelial cell, this produces antimicrobial peptide. So that peptide is there in the gut. This is goblet cell, which secretes mucus. Okay, and this is called another epithelial cell. So they line up this way. So if you look at actual velar structure look like, this is taken from a mouse. So these are small intestinal cells, right? These are the epithelial cells, and this is where the blood circulation in the middle. The blood cells are here, and these white areas, these are actually the goblet cell. Okay, and if the mucus come out, you can see this scar when mucus release, and this is when the pathogens get in. A lot of those when they are in this lumen. This is electron microscopy showing the mucus getting out of the goblet cells, and this is mucus getting out, more enlarged picture, and these are the tight junction. These are the nucleus. Okay, so that gives you some idea how the epithelial cells look like in the gut. So if you look at the electron microscopy picture, individual epithelial cell, this is one, this is one, this is one. These are the tight junction. These are called microvilli. If you go closely, these are those different types of cells or the proteins holding these epithelial cells together. And this is what I was giving you an example, that these are the proteins like a stitch, holding them together. Okay, if you pull that, it opens up, and that makes it leaky gut if this barrier opens up. Okay, can you see the connection now the leaky gut. So this junction opens up. So anything in the lumen, the microbes, the metabolic byproducts, or the food component, any of the stuff which should not be getting into your system, but if this barrier opens up or breaks down, the food or the toxic molecules can enter our body. And that's what is the part of the leaky gut system. So in order to understand gut health, you have to understand the architecture of the cells and what proteins are controlled. So all of these proteins you are seeing this is holding them together. If something happens to these proteins, your gut is not functioning properly. Okay, that's what makes it leaky gut, and all those diseases we talked about, these are contributed by the leaky gut syndrome. So that's why we talk about this physical. So literally this is the claudin protein holding them together, occludin, myosin, myosin light chain kinase, E-cadherin, all of these holding these tight junctions together. Okay, and in the laboratory, these are some of these pictures from our lab. You can stain them and you can find out Jo1 is outside. These are the E-cadherin protein in this lamina propria. This is the area called lamina propria. You can see them very clearly, all those proteins, how they are working. If something happens to these proteins in the leaky gut system, and then this protein disappears, so it becomes leaky gut. I'll show you some of them later on. So anyway, so the mucous membrane, these are also part of the mucous membrane, and they provide the protection. These are more pictures. You can see this actual tight junction area, and these are highly magnified. You go close to the individual, the cellular level, and these are the microvilli. So you saw the previous picture also. So this mucous membrane play an important role against immunity, right? We're supposed to be until 12:30. Okay, so just a little bit over now. I'll stop it soon. So the mucous membrane, you got the epithelial cells and these toll-like receptors, different types of molecules, and this Paneth cells, antimicrobial. So they all play a role in providing protection. These are part of the innate immune system. Okay, and this is the leaky gut I was mentioning. This is a review article came out 2021. So look at this, what happens. So this is epithelial barrier damage. Activated immune cells can migrate and contribute to inflammation in other organs. So this is what the Parkinson's disease or Alzheimer's disease you are mentioning, and this is the clear linkage they are establishing with the leaky gut. Let's look at this. So epithelial barrier, you saw it, right? And so this barrier damage, if the barrier damages, and then the microbes, so the microbial components can pass through this barrier, right? This is what we are showing. Then it activates immune system. And so the overall, the byproducts of this immune activation or the microbial components, they can activate immune system in a manner that can affect the brain. You see this one? Also joint. And so all those diseases now we are linking to the leaky gut syndrome. So everything starts in the gut. Got it? And that's why we have to really understand how the epithelial barrier works. How can you maintain the gut health so that you don't have this opportunity to see this. This came out of Nature Review Immunology. You can study this. And there are a lot more work being done to understand how leaky gut works. So essentially you just open it's like a floodgate, right? When there is a lot of flooding happening, the river bank breakdown, what happens? The water rush in, right? And then it's completely wipe out the village or villagers. And so leaky gut is almost like that. Because in our gut, all those microbes or their byproducts, all those toxic materials, we know we have to discard them in the morning, right? But if part of those getting into a blood circulation, think about what it can do. Okay, so our break in innate immune system, that's what is happening. Okay, I will stop here. I know any questions on this, what I just discussed? Any questions, anything? First of all, you got a clear idea now, by now, that what is natural immunity and how this natural or innate immune system breaks down and then the disease can happen, right? We haven't touched the adaptive yet, which is more vaccine, which is more long-term effect, but this is more immediate effect. And to many people, natural immunity is most important. We need to understand that first in order to provide a better understanding against any kind of chronic diseases or infectious diseases or even all those major problems we are talking about. Okay? All right, then go for break now, and I think I will see you either Monday or Tuesday, right? I don't know, this afternoon.